Kia ora team, welcome to the 2.5 Genetic Variation and Change 4-credit external video series. This is video 14. In this video, we're going to explore two important processes that change allele frequencies in gene pools: gene flow and genetic drift. By the end of this lesson, you should be able to explain how gene flow changes allele frequencies and affects diversity, explain how genetic drift has different effects on small and large populations, and explain the founder effect and the bottleneck effect as special types of genetic drift. Let's get started. Previously, you learned that genes are sections of DNA that code for proteins, and they come in different versions called alleles. Individuals inherit one allele from each parent for each gene. A population is a group of organisms of the same species living in the same area and capable of interbreeding and producing fertile offspring. You also learned that a gene pool is the sum of all alleles present in a population, and an allele frequency is the proportion of a particular allele in the gene pool. When we study evolutionary processes, we track and analyze the change of allele frequencies in gene pools over generations. In the previous video, we covered natural selection and mutation, and in this video, we're going to discuss the final two evolutionary processes that cause changes to the allele frequency in a gene pool. That's gene flow and genetic drift. Migration is when individuals move into or out of a population, and for migration to actually change allele frequencies in the gene pool, immigrating individuals have to successfully interbreed with individuals in the new population. If the immigrating individuals don't reproduce, then they don't pass on their alleles and there's no gene flow. So, we define gene flow as the movement of alleles into or out of the population through successful reproduction after migration, and this could potentially result in changes to allele frequency. Let's look at this simplified example. Before immigration, the little a allele was fixed in population 1, meaning it was the only allele in the gene pool and had an allele frequency of 100%. After immigration and successful reproduction, population 1's gene pool has gained a new allele, and the little a allele is no longer fixed. The big A allele's frequency is now 9%, and the little a allele's frequency is now 91%. So, immigration is when individuals move into a population, and if they reproduce, they bring in new alleles to the gene pool of the new population. This increases the genetic biodiversity of the new gene pool, and it may increase the allele frequency of certain alleles. It's important to note that gene flow has a bigger effect in small populations. Imagine a very small population of individuals with only one individual carrying a rare allele. If that individual emigrates, the allele is gone from the original population's gene pool, and the genetic diversity would be reduced. In contrast, in a large population, it's much less likely that one emigrant will carry the only copy of an allele. So the bigger the population, the smaller the impact each migrant has on allele frequencies. Another important point is that gene flow tends to make populations more genetically similar to each other because alleles are shared back and forth. If two populations are exchanging individuals regularly through migration, their gene pools mix and they're less likely to diverge and become different species. But if there's no migration and no gene flow between two populations, the populations may evolve separately and become genetically distinct over time. Let's look at a unique New Zealand example. The northern royal albatross is a great example of migration and gene flow because they breed on small, isolated islands but can travel enormous distances across the Southern Ocean in search for food. Most young birds ultimately return to the colony they were originally from to breed, but some disperse to other colonies to breed. There are only 2 nesting sites worldwide. 1% of all northern royal albatrosses are located in Taiaroa, Dunedin, while the other 99% of all breeding pairs are located on the Chatham Islands. If a young albatross from the Chatham Islands joins the Taiaroa Head colony and breeds there, it could bring new alleles to the Taiaroa Head gene pool. This gene flow increases genetic diversity in the Taiaroa Head population, reducing the risk of inbreeding, because remember that Taiaroa Head population is very small. At the same time, if a young albatross from the Taiaroa Head colony leaves and emigrates to breed on the Chatham Islands, it could bring new alleles to the Chatham Island gene pool. Over time, this gene flow, this back and forth, helps keep allele frequencies similar across these two colonies and prevents populations from becoming too genetically distinct. But these colonies aren't affected by gene flow equally because they greatly differ in their size. Small, isolated colonies, like the one at Taiaroa Head, is more at risk of loss of genetic diversity if there's no gene flow between the two populations. Gene flow from other colonies is vital for small populations' long-term survival because it replenishes alleles and reduces the effect of genetic drift. Without gene flow, small colonies like the ones at Taiaroa Head could lose rare alleles and become genetically bottlenecked, making them less resilient to disease and environmental change. Now let's move on to genetic drift. Genetic drift is the random change in allele frequencies in populations due to chance. These random changes cause allele frequencies to fluctuate, increase or decrease in numbers, and they can even cause alleles to disappear entirely from a gene pool just through random sampling of which individuals breed. It's important to understand that genetic drift happens in all populations. But random fluctuations in allele frequencies caused by genetic drift has the greatest effect on very small gene pools when the population is or becomes small. In small populations, relatively small changes in allele numbers can have a big impact on the allele frequency in the total population. Now, while I discuss this slide, please remember that we are going to assume there's no natural selection. That is, there's no environmental factor acting as a selection pressure on these alleles, and allele fluctuations or trends are not because of beneficial or harmful alleles. This slide is purely about genetic drift, which is the random change in allele frequency due to chance. When populations are large, allele frequencies tend to remain stable from generation to generation. In this example, we can see that a large breeding population of 2,000 individuals, genetic drift has little effect because the random changes in allele frequency tend to average out, and the trend line, as you can see, is very stable. When populations are small, any random changes to allele frequencies tend to have a larger proportional effect. In this example, we can see that in a smaller breeding population of 200, changes in allele frequencies tend to have more severe fluctuations on this graph. But overall, the trend line could still be quite stable. However, when populations are very small, changes in allele frequencies fluctuate so much that they drift into an increasing or a decreasing trend. In this example, we can see that a very small breeding population of just 20 individuals leads to extreme fluctuations that drift into this decreasing trend in allele frequency. It ultimately leads to an allele loss. These graphs clearly show how larger populations can maintain more genetic diversity because allele frequencies are able to stay stable despite small genetic drift fluctuations. It clearly shows how very small populations are less able to hold onto that genetic diversity because genetic drift causes huge fluctuations in allele frequencies, and that allele loss is quite probable. I've already mentioned that genetic drift happens in all populations, but there are also two special types of genetic drift that you have to know about: the founder effect and the bottleneck effect. First up, let's discuss the founder effect. This is a special type of genetic drift caused by colonization. The founder effect occurs when a small population of individuals migrate away or become isolated from a large population. The smaller founding population only carries with them a small sample of alleles from the larger original population. So the range of alleles and the frequency of alleles present in the small founding population is unlikely to be representative of the original population. Some alleles will be less frequent, others will be more frequent, and some alleles in the larger original population may not be present in the founding population at all. As a result, the descendants of the founding population may have very different allele frequencies compared to the original population. In this hypothetical situation to the right, a small randomly selected population of beetles is blown off to a neighboring island where they establish a breeding population of 8 individuals. When we compare the allele frequencies between these two populations, we can see that the smaller founding population and the larger mainland population have quite different allele frequencies. Because the founding population is so small, it will be more affected by genetic drift. Any random changes to allele frequencies will tend to have a larger proportional effect on the smaller founder population, and as a consequence, this founding population may evolve in a different direction than the original population because the environmental conditions for the isolated founding population may be different. In some cases, certain alleles could be missing altogether from individuals in the isolated founding population, meaning future generations of the founding population won't have this mainland allele. The founder effect is particularly relevant to many native species of New Zealand. Many of our native species arrived to our country from Australia or New Caledonia in small numbers by chance, such as the silvereye, the white-faced heron, and the spur-winged plover. These founder populations were unlikely to have had the range of alleles or the frequency of alleles present in the populations they originated from in Australia and New Caledonia. When you combine this with the fact that the founder populations are in a new environment, New Zealand being its own environment, with different selection pressures acting on the new gene pools, you can see that the founder population has the potential to become quite different from the original populations. The bottleneck effect is the second special type of genetic drift, but this time around it's caused by a population crash. The bottleneck effect occurs when populations are suddenly reduced to very small numbers due to a catastrophic environmental event or human action. In either case, individuals are indiscriminately killed off or removed regardless of their genetic makeup. It's important to understand that the population crash is not selective. It affects all phenotypes randomly. So the survivors' alleles are a random subset of the original gene pool. And the gene pool after a bottleneck effect may not have a representative sample of the alleles in the pre-catastrophe population. It's likely that the range of alleles will decrease and the frequency of alleles will change. And even if the population numbers recover to its original size or even larger, the bottlenecked population will have reduced genetic diversity. The small surviving population is often affected by inbreeding and genetic drift, which may cause further changes to allele frequencies or even allele loss. This diagram clearly demonstrates the effect of the bottleneck effect on genetic diversity here at the top and population numbers here at the bottom. Pre-catastrophe, the insect population was large and genetically diverse. Then a catastrophic event takes place, and the population numbers crash to near extinction. This small population will be significantly affected by genetic drift, so random allele fluctuations due to chance, and also inbreeding, mating with close relatives. So even when the population numbers recover and bounce right back to the pre-catastrophe numbers, the gene pool's genetic diversity will be severely limited. The gene pool of the post-catastrophe population will not be representative of the pre-catastrophe population's gene pool. Cheetahs almost went extinct at the end of the last ice age. Their population crash was so severe that it's thought that only a single family group survived. This would have led to inbreeding and explains the lack of genetic diversity found in modern-day cheetahs today. Their lack of genetic variation has led to a number of features that threaten the survival of cheetah species today. This includes sperm abnormalities, reduced fertility, high cub mortality, and sensitivity to disease. Since that genetic bottleneck, there's been insufficient time for random mutations to accumulate and introduce new genetic variation. So even today, their genetic diversity is incredibly low. This diagram closely represents the bottleneck effect that happened in cheetahs. So the original cheetah gene pool was made up of offsprings of many lineages. Then during the Ice Age extinction event, only a single family group of cheetahs from one of the lineages survived and reproduced. Eventually, the population numbers increased, but because only closely related individuals survived, there was a lot of inbreeding. The consequence of this inbreeding is the lack of genetic diversity we see in the pool of modern cheetahs today. In fact, all cheetahs today can be traced back to that one lineage that survived the bottleneck event. So when the population recovers, it will have reduced genetic diversity due to inbreeding and genetic drift. And as a result, we see all of these harmful features to the cheetah's survival, such as reduced sperm quality, increased infant mortality, and reduced fertility. So that's all for this lesson. By now you should be able to explain how gene flow changes allele frequencies and affects diversity, explain how genetic drift has different effects on small and large populations, and explain the founder effect and the bottleneck effect as special types of genetic drift. Thank you for watching and see you in the next video.